Hey students, this is your Nandini ma'am. Today we are going to resume our reading from the topic enzymes, and the main topic was cell functions. So let's get started. Enzymes are protein molecules that control all metabolic reactions in living cells. Without enzymes, the reactions that occur in cells would be so slow so as to hardly proceed at all. Enzymes act as biological catalysts, controlling the rate of each step of the complex chemical reactions that take place in cells. Catalysts are chemical substances that can accelerate chemical reactions, are unchanged at the end of the reaction, and can be reused many times. Metabolism is the sum of all chemical reactions occurring within a living organism. Over 1,000 different reactions can take place in each cell. A specific enzyme catalyzes each of these reactions. There are as many enzymes in living organisms as there are types of chemical reactions. Atoms and molecules are in constant motion and colliding. If the reactants are supplied with enough energy, such as heat, to break their bonds, then the reaction will proceed and products will be formed. The minimum amount of energy required to start the reaction is called the activation energy. Enzymes speed up the reactions by lowering the activation energy required for the reaction. They do this by combining with the reactants and holding them in a way that makes the reaction more likely to occur. Properties of enzymes. Enzymes are composed of protein molecules that are often highly folded to create a particular chemical shape. The surface of the enzyme with a specific shape is called the active site. It is here that the reactants, also known as the substrates, in a chemical reaction temporarily bind. When this occurs, a substrate enzyme complex is formed and the reaction proceeds. The products of the reaction are then released from the active site of the enzyme. The enzyme remains unchanged. It is again available for the same process to occur. An enzyme is specific for a particular reaction as the shape of the active site will accommodate only substrates that match that particular shape. Models of enzyme activity. Two models have been developed to try to explain how the substrate binds at the active site to form the substrate enzyme complex. First, a lock and key model was proposed. It was thought that the active site is rigid and the small substrate molecule is reciprocally shaped and fits exactly into the active site like a key into a lock. Once this substrate enzyme complex has formed, the close proximity of the molecules allows the reaction to be rapidly catalyzed and the products of the reaction are released. To validate this model, predictions were made and tested. The results led to the proposal of the currently accepted amended version of the model known as the induced fit model. This model is based on the realization that proteins are not rigid. Evidence suggests that the binding of a substrate to the active site of an enzyme induces the enzyme to alter its shape slightly to fit more tightly around the substrate. Factors affecting enzyme activity. Enzymes require particular conditions in order to function at their optimal efficiency. Any change to these conditions means the enzyme will either work very slowly or not at all. Enzymes are temperature sensitive. Enzymes within cells function best at the body temperature of the organism in which they occur. In most living things, enzymes function normally at temperatures up to 40 degrees Celsius. Some organisms that live in hostile environments have optimal temperatures that are much lower or much higher than this. In humans, the optimal temperature for the functioning of enzymes is 37 degrees Celsius. As the temperature increases, the rate of reaction increases until the optimal temperature is reached, at which point the enzyme activity is at its maximum level. Above this temperature, the enzyme activity slows until it stops altogether at temperatures of approximately 50 to 60 degrees Celsius. At high temperatures, the motion associated with heat energy can make the protein structure bend and flex so much that it changes the shape of the active site. This renders it incapable of accommodating the substrate that would normally bind there. Therefore, the enzyme will not function as it should. The heat has caused the enzyme to denature. This change in the structure and shape of the molecule due to excessive heat is irreversible. Excessive cold also causes enzyme to change shape and its functioning to slow down or stop. This change in shape due to excessive cold is, however, often reversible. Enzymes are pH sensitive. pH is a measure of the acidity or alkalinity of a substance or solution. Acidic solutions have pH values below 7, while alkaline solutions have pH above 7. Neutral solutions have a pH value of 7. Each enzyme has its own narrow range of pH within which it functions most efficiently. Levels of alkalinity or acidity outside the optimum pH for an enzyme have a similar effect to that of temperature change. They alter the shape of the enzyme and slow down or stop its function. Extremes of pH like temperature cause the enzymes to denature. Within cells, most enzymes function at or near neutral, but enzymes in the digestive tract function in an acidic or alkaline medium. For example, the protein digesting enzymes, pepsin and renin, found in gastric juice in the stomach, function best in a strong acid or low pH. Substrate concentration and enzyme activity. The rate of an enzyme-controlled reaction is affected by the concentration of the substrate. The higher the substrate concentration, the greater the rate of enzyme reaction until all available enzymes are being used to catalyze reactions. This point is called the saturation point. Increasing the substrate concentration beyond the saturation point will not increase the rate of reaction since all enzymes are working at their maximum turnover rate and will have to be reused to act on the additional substrate. The only way to increase the reaction rate would be to increase the enzyme concentration. Investigation 3.3, a practical investigation to model the action of enzymes in cells. Catalase is an enzyme present in all living things. It is responsible for catalyzing the breakdown of hydrogen peroxide. Hydrogen peroxide is produced as a result of cellular respiration within the cell. If it is allowed to accumulate, it will be toxic to the cell and cause its death. When hydrogen peroxide is broken down, it forms the non-toxic substances, oxygen and water. This investigation models the action of enzymes in cells to justify the substrate enzyme binding action and reusability of enzymes. It investigates the effect of substrate concentration on the activity of an enzyme. Models make predictions to explain how things work. In this model, if enzymes bind with substrates to catalyze a reaction, then an increase in substrate concentration should result in the saturation of enzymes at a particular substrate concentration. If further substrate is added beyond the saturation point and the enzyme concentration is kept the same, there will be no further increase in the rate of reaction. Hydrogen peroxide will represent the substrate and catalase in potato is the enzyme. As oxygen gas is a product of this reaction, the activity of the enzyme is determined by measuring the height of the bubbles produced in the hydrogen peroxide. Similar investigations can be performed with hydrogen peroxide and potato to determine the effect of temperature and pH on the activity of an enzyme. Aim: to model the action of enzymes in cells based on the substrate enzyme binding action and reusability of enzymes. To investigate the effect of substrate concentration on the activity of an enzyme. Hypothesis: the activity of the enzyme will increase as the substrate concentration increases until it reaches saturation point, after which the activity of the enzyme will remain constant. Materials: 7 test tubes of the same size, test tube rack, hydrogen peroxide 6%, 2 into 10 ml measuring cylinders, cork borer or blender, 30 centimeter rule, marking pen, potato, labels, stopwatch. Risk assessment. Hydrogen peroxide. Toxic if ingested, eye and skin irritant. Wear safety goggles and disposable gloves. Do not ingest. Potato. Can be a skin irritant. Wear disposable gloves. Method. Label the 7 test tubes with the numbers 1 to 7. Measure the quantities of hydrogen peroxide and water specified in Table 3.9 using the measuring cylinders and place them in the appropriately numbered test tube. Mark the level of the liquid in each test tube using the marker pen. Using a cork borer, obtain 6 cylinders of potato of the same diameter. Cut the potato cylinders so that they are of equal length. Note, an alternative way to prepare the potato is to puree cubes of potato with a small amount of water in a blender. A few ml of this pureed potato would then be measured into the test tubes instead of the cylinders of potato. Place a cylinder of potato in each of the test tubes numbered 2 to 7 and allow the reaction to proceed for approximately 5 minutes. At the completion of this time, place another mark on the test tube to indicate the maximum height to which the bubbles reached. Measure the difference between the level of the liquid and the height to which the bubbles reached in each test tube. Record your results in a result table like Table 3.10. Compare your results with those of the other groups in the class. Graph the results obtained using a line graph. Remember that the independent variable goes on the x-axis and the dependent variable is on the y-axis. Discussion. Identify the enzyme substrate in this model of enzyme activity. Describe how test tubes 1 and 2 model the action of enzymes in cells. Was this a valid model? Justify your answer. Identify the following variables: independent variable, dependent variable, controlled variable. Why was test tube 1 with no potato added used? Discuss the validity of the experimental design. Describe the trend obtained on your graph. Is the hypothesis supported by these results? Compare your results with other groups in the class and comment on the reliability of the investigation. Conclusion. Write a conclusion that relates the aim of the investigation to the results of your investigation. Design your own investigation. Investigate the effect of pH or the effect of temperature on enzyme activity. This can be undertaken using the same substrate and enzyme and a similar method, or you may wish to research methods using other readily available enzymes and substrates. Define a research question and hypothesis for your investigation. Follow the steps outlined in Chapter 1 to assist you with your investigation. Key concepts. Cellular products and wastes must be removed from the cell to maintain efficient cell functioning. Enzymes or biological catalysts are proteins that control cellular reactions. In enzyme catalyzed reactions, the substrate attaches to the shape on the surface of the enzyme, the active site, and forms a substrate-enzyme complex. The reaction then occurs and the products are released. The enzyme can be reused for the same reaction. There are two models of enzyme action, the lock and key model and the induced fit model. The activity of an enzyme increases as the temperature increases until the optimal temperature is reached. With further temperature increases, the enzyme activity decreases and then stops completely when the high temperature destroys the structure of the enzyme. Each enzyme has an optimum pH at which it functions most efficiently. Enzyme activity is affected by the substrate concentration. As the substrate concentration increases, the activity of the enzyme increases until all the enzymes are saturated. After this, further increases in substrate concentration will not lead to increase in enzyme activity. Biochemical processes in cells. Eukaryotic cells contain many membrane-bound organelles that contain specific enzymes that catalyze specific biochemical processes. For example, Chemical reactions in lysosomes break down compounds brought into the cell by using strong digestive enzymes in an acidic environment. Having the lysosome enclosed in a membrane prevents the enzymes from destroying the cell. These reactions within cells require energy, which is provided in the form of ATP. The two major biochemical processes that cells carry out are photosynthesis in plant cells and cellular respiration in living cells. Energy transfer between reactions. Provision of energy is vital to ensure that all essential life processes take place. All cells use glucose as the primary source of energy to drive the thousands of chemical reactions that occur constantly in each living cell. When glucose is broken down in the process of aerobic cellular respiration, the large amount of energy contained within it is released. This released energy is trapped and stored in small packets in many high energy molecules called adenosine triphosphate or ATP. The cell assesses the energy contained in ATP to power all cell functioning. ATP is a small mobile molecule that can deliver the energy to wherever it is required. ATP is composed of a complex molecule called adenosine. Adenine attached to a ribose sugar group that in turn attaches to three phosphate groups. This is where the name adenosine triphosphate comes from. The bond that attaches the third phosphate group is the high energy bond. When energy is required by the cell, this bond is broken and the energy it contains is released. ADP and a free phosphate group are formed. To store the energy produced by the cell, a free phosphate group is attached to the ADP molecule by a high energy bond, forming ATP. The storage and release of energy is a cycle between ADP, free phosphate groups, and ATP, as shown in figure 3.34. Photosynthesis. Photosynthesis is the process by which plants utilize light energy, usually from the sun, which is trapped by chlorophyll. It uses this energy to break apart water and carbon dioxide molecules and build them up into oxygen, energy storing glucose molecules, and water molecules. So, photosynthesis can be summarized by the word equation: carbon dioxide plus water in the presence of light energy and chlorophyll gives glucose plus oxygen. The process of photosynthesis is summarized by the journal equation above. The actual process of photosynthesis occurs in two stages, each consisting of a series of many chemical reactions that take place in the chloroplasts of green plants, plant cells, and the cells of many photosynthetic bacteria. Phase 1. Light-dependent stage. The light-dependent stage, photolysis, occurs in the grana of chloroplasts. It involves the absorption of light energy by the chlorophyll in the thylakoid membranes of the chloroplast. This energy is used to split the water molecules into hydrogen ions and oxygen. The oxygen is released into the atmosphere and the hydrogen ions are carried to the next phase. ATP is also formed at this stage. Phase 2. Light-independent stage. The light-independent stage, carbon fixation, occurs in the stroma of the chloroplast. It involves combining carbon dioxide with the hydrogen ions produced in the light-dependent stage to form glucose. The ATP formed in the light-dependent stage is required to produce the energy for this reaction. No light is required at this stage. Glucose produced by the photosynthesis can be converted by plants into complex carbohydrates, lipids, and or proteins. These end products are stored by the plants and are the source of organic nutrients not only for the plants themselves, but also for the consumer organisms that eat the plants. Key concepts. Energy in cells is transported within cells by small and mobile ATP molecules. ATP stores energy in a high energy bond that attaches the third phosphate group to the ADP molecule. When energy is required, the high energy bond is broken, releasing energy, a phosphate group, and ATP. Photosynthesis is the process by which plants use light energy trapped by chlorophyll to break down water and carbon dioxide molecules and build them up into oxygen, glucose, and water molecules. Photosynthesis is a series of smaller reactions occurring across two stages. Light dependent stage occurs in the grana where light energy splits the water molecules into oxygen and hydrogen. Light independent stage occurs in the stroma where carbon dioxide and hydrogen combine to form glucose molecule. Cellular respiration. All organisms break down glucose as a source of energy to drive cellular metabolism. Glucose can be broken down either in the presence of oxygen or in the absence of oxygen. Different products and amounts of energy are released in each of these processes. Anaerobic cellular respiration. Many organisms, such as some bacteria and archaea, live in environments lacking oxygen. Cells such as muscle cells sometimes cannot get enough oxygen, but still are able to release energy via an anaerobic pathway in the cytosol of cells. Two commonly used biochemical pathways enable energy to be produced using molecules other than oxygen: alcohol fermentation and lactic acid fermentation. In alcoholic fermentation, yeast, many bacteria, and plants carry out anaerobic cellular respiration. This process involves the breakdown of glucose in the absence of oxygen to form ethanol and carbon dioxide. Two molecules of ATP are produced as a result. Humans make use of this process in the production of beer, wine, and bread. Lactic acid fermentation is carried out when an organism cannot produce enough energy by aerobic cellular respiration. This occurs in periods of strenuous exercise when our bodies cannot deliver enough oxygen to the muscles to produce the energy required. In lactic acid fermentation, one molecule of glucose is broken down in the absence of oxygen to produce two molecules of lactic acid as well as two molecules of ATP. Once the exercise has finished, there will be more oxygen available. The lactic acid changes to a compound called pyruvate and is converted by aerobic respiration into carbon dioxide, water, and energy, ATP. Aerobic cellular respiration. Aerobic cellular respiration is a chain of many biochemical reactions that occur in the cells of the organism in the presence of oxygen. It is generally summarized as follows to depict the main changes that occur during respiration. There are at least 20 separate reactions, each catalyzed by a specific enzyme that makes up this overall pathway. The first step in the biochemical pathway to break down glucose is the process of glycolysis. This process occurs in the cytosol of the cell and involves the breakdown of glucose into two pyruvate molecules. This is accompanied by the release of energy in the form of two molecules of ATP. This overall reaction is actually a series of 10 smaller reactions, each catalyzed by a specific enzyme. The two molecules of pyruvate formed from one molecule of glucose then enter the mitochondria of the cell, where the rest of the series of reactions that make up aerobic cellular respiration occur. Each of these reactions is catalyzed by a specific enzyme and accumulates in the production of more energy, carbon dioxide, and water. The energy from these reactions is released in the form of 34 ATP molecules to make a total of 36 molecules of ATP produced for the breakdown of every glucose molecule. This process is summarized in figure 3.37. Photosynthesis and aerobic cellular respiration. Photosynthesis and aerobic cellular respiration are related. When you look at the journal equations for the photosynthesis and aerobic cellular respiration, it appears that these processes may be the reverse of each other. That is not the case because they are each made up of series of chemical reactions that are very different. Photosynthesis and aerobic cellular respiration are, however, closely related and interdependent. The products of photosynthesis are used in the process of aerobic cellular respiration. Similarly, the products of photosynthesis are used in the process of aerobic cellular respiration. Similarly, the products of aerobic cellular respiration are used in the process of photosynthesis. In cells that contain both chloroplasts and mitochondria, the two processes will occur in the same cell. In cells that do not contain chloroplasts, such as plant root cells, the products of photosynthesis must be supplied to the cells for the process of cellular aerobic respiration to occur. Removal of cellular products and waste. The products of the reactions in the cell that are not required by the organism are called waste. They need to be transported to other cells and or need to be removed for efficient cell functioning. Removal from the cell will usually occur by diffusion through the cell membrane. Oxygen and carbon dioxide gases are easily removed from the cell by diffusion through the cell membrane. Any excess water that is not required by the cell moves out of the cell by osmosis. Wastes formed from the breakdown of protein also leave the cell by diffusion through the cell membrane. Waste chemical products that dissolve in water are removed from cell at the same time as the water. Oily, fat-soluble substances are attached to small, electrically charged molecules by the cell to make them water-soluble so that they can be removed when the water diffuses out. When parts of the cells break down, wear out, or die, they also need to be removed. Lysosomes, when contain digestive enzymes, are responsible for breaking down these old cell parts and other cellular waste. Any of these wastes that can't be eliminated in this way by the lysosome are packaged and removed by exocytosis. Proteins such as hormones and enzymes and other substances such as mucus are produced by an animal cell. These cellular products are weighed, packaged into vesicles and leave the cell by the process of exocytosis. Wastes that leave a cell are said to be excreted from the cell. Useful products that leave a cell to be used elsewhere are said to be secreted. Key concepts. When glucose and oxygen combine in the process of aerobic cellular respiration, energy is produced along with water and carbon dioxide. This process is made up of many reactions. The energy produced is stored in ATP molecules. The first step of this process occurs in the cytoplasm and is called glycolysis. In this step, one glucose molecule is broken down into two pyruvate molecules and 2 ATP. The second step in the reaction occurs in the mitochondria where pyruvate in the presence of oxygen is broken down to release carbon dioxide, water and 34 ATP. A total of 36 ATP molecules is produced for every molecule of glucose broken down in aerobic respiration. Anaerobic cellular respiration produces ethanol and carbon dioxide in bacteria, yeast and plant cells, and lactic acid in animal cells. 2 ATP molecules are produced in this process for the respiration of each molecule of glucose. Wastes are the products of cellular reactions that are not required. Many of these wastes can be removed by simple diffusion through the cell membrane. Other waste can be removed by exocytosis or destroyed by the enzymes present in lysosome. So with this, we finish off our chapter. Happy reading and enjoy your studies.